Hi, everybody, and welcome to the presentations tonight. My name is Ned Katire. I'm a pediatrician and the medical advisor for Southwest Pennsylvania Environmental Health Project. EHP supports residents of Southwestern Pennsylvania and beyond who believe that their health has been or could be impacted by unconventional oil and gas development. Information about EHP can be found at www.environmentalhealthproject.org, and I'll put that in the chat in a few minutes. We have a fascinating and important program for you this evening. Our two distinguished panelists will be talking about endocrine disrupting chemicals and shale gas development, which includes the risk of exposure to harmful chemicals at the wellhead and beyond. At compressor stations and pipelines, at processing facilities and export sites, at ethane cracker plants and other processing, other plastics and petrochemical facilities, and in the environment, as the toxic components and the additives to plastics slowly break down and enter our air and our oceans, our soil, our food, and eventually our bodies. Both speakers tonight, Dr. Chris Kassotis and Dr. Laura Vandenberg, conduct remarkable research into endocrine disrupting chemicals, what they are, where they come from, how they operate to disrupt critical hormonal functions in the body, and how they can make us sick. Last year, Dr. Kassotis and Dr. Vandenberg collaborated on endocrine disrupting impacts of chemicals used in unconventional oil and gas development and found in the industry's wastewater. The results of their study, which involved experiments on lab animals and human tissue culture cells, were rather disturbing to me as a pediatrician. In fact, the human health and ecological impacts from fracking and cracking and on through the life cycle of plastics should disturb every pediatrician, every medical provider, and every parent. I can't recommend highly enough the report co-authored by Dr. Vandenberg and published by the Endocrine Society in December on plastics, EDCs, and health. The opening sentence in the foreword prepares the reader for an inevitable reckoning. I quote, "Chemical additives in plastic and the threat they pose to human health and the environment is an emerging issue of global concern that is garnering increasing attention as society is beginning to address the worldwide plastic pollution problem." Well, thanks to the work of our two presenters tonight, I can say confidently that the problem of endocrine disrupting chemicals found in fracking pollution, in the plastic products that we're also fond of, and in the enormous waste stream those plastics generate, this is no longer an emerging issue of global concern. Dr. Kassotis and Dr. Vandenberg have our attention. We would be wise to heed the warnings emanating from the results of their research. So our first presenter tonight is Chris Kassotis, PhD, an assistant professor in the Institute of Environmental Health Sciences and Department of Pharmacology at Wayne State University in Detroit. Dr. Kassotis is head of the Kassotis Lab at Wayne State. His research is focused on identifying and characterizing novel endocrine disrupting chemicals from diverse sources, including unconventional oil and gas operations, and evaluating the potential health effects from these exposures. You can follow Chris's exciting work at www.kassotislab.org. Tonight, Chris will be presenting From Fracking to Fat Cells, How Unconventional Oil and Gas Production May Influence Metabolic Health. Chris, please take it away.
Thanks so much for the very kind introduction and for the invite to come and talk with you guys today. And it's of course, even bigger delight because I have the opportunity to present next to Laura, which is always a great deal of fun. So today I'm going to tell you a little bit about one of the projects that has kind of become a passion for me over the years. So I started this back in oh gosh, I'm going to start dating myself. Back in 2010, when I started my PhD, and it quickly became my dissertation work, and it has continued with me through my postdoc and now into my independent faculty position here at Wayne State. So I'm excited to talk to you about that, and give a little bit of an introduction to endocrine disruptors if you're not familiar. So if you are here, this is probably no surprise to you. So hydraulic fracturing, right? So this is the general schematic of the process. So it's involving so we're drilling a well bore down to what we would think of as a previously inaccessible geological layer far underground, although I'll say this varies quite a lot across regions. And then the drillers are able to turn their drill bit horizontally, and they can drill up to a mile or more through that shale or coal bed gas layer. And then that's followed, so that kind of directional drilling is followed by the hydraulic fracturing process, which is the high pressure injection then of water and chemicals and proppant that serves to fracture that target layer. The proppant kind of wedges itself into those fractures, and then it allows for the oil gas to come up and be collected from the well. So as I mentioned, a lot of chemicals are used in this process, although as industry will say, this is often, a very low percentage. When we're talking about several million gallons of fluids being used to fracture an individual well, it's still an awful lot of chemicals being used at every individual site. So this schematic overviews a little bit of potential routes of contamination. Of course, we can have spills of chemicals or fluids at the well pad prior to injection. During the mixing process, during the injection process, there are certainly potential for contamination of groundwater due to the casing of the well. There have been some descriptions of potential for migration of fluids that have been injected deep underground to eventually migrate back up into the groundwater of the region. And then, of course, maybe the biggest contamination source is then what happens to all these fluids after the fact, right? And so that also depends on geology. In some areas, you're going to recover a lot more of the injected fluids back at the surface. And in other areas, you're going to recover very little. But what do we do with those, right? So in a lot of places, we are transporting them to evaporation pits and essentially evaporating the chemicals. And then often they'll just turn soil over these pits and leave that there. What's become more and more common is the use of injection wells. And so we're, in that case, injecting into more porous underground formations that can soak up some of that fluid. Of course, wastewater treatment plants and the potential to treat some of that residual wastewater has been explored in other areas. Certainly, we're sending a lot of the clippings and other materials to landfills across the country. And then depending on, a lot of your state guidance, some places will allow things like beneficial reuse. So we've seen that in California where wastewater is being used for agricultural watering. And then things like dust suppression, where it's actually sprayed over roads to keep dust down, which is potentially then spreading contaminants out onto roads that people are walking and driving and moving down. So we got into this really largely due to the work of Theo Colborn and the Endocrine Disruption Exchange. And so they were a nonprofit group in Colorado that was really interested in endocrine disrupting chemicals, and largely due to landowner concerns, started investigating unconventional oil and gas or fracking as a potential health threat to people living in the area. And so one of the things that they did that they do so well is they did a really nice systematic search of the literature for health outcomes associated with the chemicals used in the process. And at this point in time, there really wasn't a lot known about which chemicals were injected into wells. And so they spearheaded some chemical analysis by some contract labs, and built a database, what I think the earliest database of chemicals used in the process. And so at this point, they had about 350 chemicals that they had found. And they found that about 40% of those, there was some suggestion in the literature could be endocrine disrupting chemicals. And so what is an endocrine disrupting chemical, right? So hormones are really important for not just early life development and, establishing all sorts of body processes, pubertal development, sex characteristics, brain development, but also just maintaining all sorts of body processes throughout our lives, right? So regulating metabolism, regulating immune function, cell proliferation and maintenance, or muscle and bone formation. And there's a lot of interplay across these receptors, right? So you might have effects of the estrogen receptor on something like bone formation, but there are other receptors that also play a role in that. And so really the effects of hormones are very far reaching. It's hard to find anything in the body that hormones are not at least involved in. And so when we start thinking about endocrine disruptors, these are any chemical or mixture of chemicals in the environment that are able to interfere with any aspect of hormone action. And so what do we mean about that, right? So generally what I'll be talking about are direct interactions. So these are what we would call receptor agonists or antagonists. And so an agonist is essentially just a hormone mimic. So this is a chemical that is able to bind to a hormone receptor in much the same way that a hormone would in the body and drive a biological effect, inadvertently, you would say. So that's kind of a receptor activator. And then an antagonist is more of a receptor inhibitor. And that's where a chemical is able to bind to or otherwise prevent the endogenous or the natural hormone from binding to the receptor and then prevent that biological effect from occurring. And of course, there's all sorts of other effects that these chemicals can have. They can affect, where these receptors occur in the body and at what levels. They can affect actual hormone levels in the bloodstream and even how the receptor responds when it binds the hormone and many other certainly mechanisms. And where are these found? All over the place, right? So certainly well described. There's a lot of attention on chemicals like bisphenol A, the phthalates, but often pharmaceuticals are designed to hit some of these endocrine targets to exert their mechanism of action. But there's a lot of industrial chemicals, metals, personal care products, a lot of pesticides, et cetera. And so then what happens, right? So then we get dysregulation or can have dysregulation of all of those pathways that I showed you before that are regulated by hormones, right? And so I'm not going to belabor this, but I will say, I will just focus the attention here that one of those is potentially contributing to metabolic syndrome. So, right, so this is obesity, type 2 diabetes, and cardiovascular disease. And so a lot of the work that I did during my PhD was looking at common use chemicals used in fracking. And so I tested a set of 24 that industry reported to be commonly used. And I did a lot of direct testing to see if they interacted with human hormone receptors. And then I followed up with a study where I exposed pregnant mice to a mixture of 23 of these chemicals, and then monitored the health of the offspring kind of throughout early adulthood. And just to give you a little idea of what that mixture looked like, so there's a range of kind of more volatile chemicals, some more semi-volatile chemicals. And so these are some of the, what industry says are the most commonly injected chemicals in a hydraulic fracturing well. And that was, quite a few years ago. And so what do we find, right? So I'm not going to belabor this, and Laura might touch a little bit more on this in the next talk. The one thing that I'll highlight here is that we saw increased body weight and birth weights in both our male and female pups who were just exposed while their mother was exposed during pregnancy. All right, so how does that happen, right? So I think everyone appreciates at this point that this is a large problem. So currently the newest numbers are 42% of the US adult population is categorized as obese. It's over 70% overweight and obese. That's 1 in 10 infants and toddlers, 1 in 5 to 19-year-olds. So this is a quite dramatic problem at this point. It contributes to a huge amount of US healthcare costs and is of course associated with some comorbidities, right? So things like type 2 diabetes, cardiovascular disease, hypertension co-occur with obesity. And one of the reasons this has gotten a lot of attention is that really interventions have produced only modest effects in most cases, right? And while most of that is relative to the US, this is of course a problem around the world. And we've seen increasing rates of obesity in almost every country that we look at. So this idea that environmental chemicals are driving some of these effects first started to get attention back in 2002. And this paper kind of challenged the idea that genetics, activity, and caloric intake were insufficient to explain the magnitude and the speed of the increasing rates of obesity in the US. And the other thing it did was it summarized the wealth of the available evidence on all sorts of environmental chemicals that could increase weight gain, right? So antibiotics, there were papers being published back in the 50s on exposing agricultural animals to antibiotics or estrogens to fatten them up, right? So there's a ton of evidence out there showing that environmental chemicals can increase weight. So most of what I'll say today is going to be surrounding this cell line. So this is a mouse pre-adipocyte cell line. So these are cells that are essentially early stage fat cells. So until they get a further push, they're still going to look kind of like a multipotent stem cell sort of characteristic. But once they get pushed, so they can never develop into any other cell type, they will only ever develop into a fat cell. And because they are so readily cultured, we have been using them for decades now to understand how fat cells develop and understanding the mechanisms through which that happens. So it's about a 2-week process to expose these chemicals and monitor fat cell development. And at the end of it, we look at the accumulation of lipids in the cell, which is what you can think of as kind of an increased fat cell size, and then an increase in the number of fat cells or proliferation. And you can kind of see that here. So these are some actual fat cells from my lab. And the yellow staining is the lipid accumulation. And you can see that we've got some blue nuclear or DNA staining, and then you often see these larger or, at varying stages of development, lipid droplets surrounding the nucleus of the cell. All right, so I'll be telling you about 2 kind of metrics here, right? So as I mentioned on the last slide, one is lipid accumulation or triglyceride accumulation. And I'll always show that on the left-hand side. And so you'll have increasing concentration on the bottom of the graph, and then increased magnitude of effect going up. And then cell proliferation on the right-hand side. So one of the things that we did during my time at Duke was we looked at the same mixture of chemicals that we had tested in mice to see, will this affect fat cell development in this cell model? And we found that yes, it does. So it's shown in blue here is our positive control, Rosiglitazone. So this is a well-described chemical that provides kind of robust fat cell development. And so relative to rosiglitazone, it certainly happens at higher concentrations and to a lower magnitude. But this 23 mix of chemicals that promoted heavier mouse pups also drives increased lipid accumulation and actually a much more significant effect on proliferation. So we got increased number of fat cells as well. Now, I'll highlight this real quick. 2 of those 23 chemicals are these ethoxylated surfactants. So these chemicals are used in unconventional oil and gas. They're present in our mixture that we've tested a couple times. They're consistently reported in the top 50 chemicals used in hydraulic fracturing, and they've been measured in unconventional oil and gas wastewater by a number of different groups now. What are they? They're really high production volume chemicals, and they're used widely in laundry detergents, hard surface cleaners, paints, cosmetics, and even some agricultural products. So we wanted to look at these chemicals a little bit more and see, could these be some of the, the bad actors here causing some of the effects that we saw in fat cell development. And so here's just a set of 6 different surfactants. And all 6 of them, shown here on the left, induce lipid accumulation or the extent of fat cell development in our cell model. And then 2/3 of them induced increased proliferation. Now, if we look at one thing we were interested in is how the size of the chemical influenced this effect. And we actually see this nice little inverted U that Laura will enjoy, I think, where we see with increasing size of the chemical, we actually see an increase in our fat cell development until you reach a certain point, and then we actually see a decrease from there. So we've got this nice inverted U here. All right, so the last thing I'll just share a little bit about is moving on to test some of these in zebrafish. And so very briefly, zebrafish. Why zebrafish? So imaging of whole animal fat or adipose in mammals is very challenging and limiting, generally low resolution, whereas you can do that very easily in zebrafish. They have 34 distinct fat cell depots in the body, and they've been clearly developmentally detailed by John Rawls' group at Duke, allowing us to easily detect changes in the normal developmental timeline. So to give you an idea of what this looks like, here is a kind of control zebrafish. So they've only been exposed to our solvent, which we're going to dilute our test chemicals in. And you can start to see here around the pancreas, a couple little, couple lipid droplets, a couple fat cells here lighting up a little bit more than the rest. Contrast that with one of these ethoxylate treated fish, and you can see there is a lot more fat in this fish, right? So you've got a huge depot here, this pancreatic and visceral depot. You've got all sorts of ocular depots. So there's just a lot more fat in this fish. So I'm not going to belabor this. I'll just say we're exposing the fish in early life. So one day after they're born, so they're still embryos at this stage until day 6. And then we age them out to 30 days. And that's when we image them to measure the growth and then the measure the adipose development in the fish as well. So this just summarizes one of the metrics of how old the fish are is just how long they are, standard length. And this is just to show you that nothing that we tested alters the length of the fish. So that kind of measure of development has not changed. So all the way on the left, you have kind of our background. This is our control group. We've got a positive control, and then we've got kind of increasing size of these ethoxylates. Whereas if you start looking at the weight of the fish, you'll see that the positive control, tributyltin, does increase the gross weight I'll say it also increases the adipose in the fish. And then you don't really see effects here at these lower size ethoxylates, but as you increase to this kind of nonylphenol ethoxylate 6 or 10, you see an increase. Of course, I couldn't make it through. You see an increase in body weight for your fish at the same size of molecule that you see an increase in our cell model. All right, so what are the take homes, right? So unconventional oil and gas production chemicals and mixtures may disrupt metabolic health. We've measured this in our cell model for lab created mixtures of common use chemicals in wastewater impacted surface water samples from drilling dense regions. We've sampled in Colorado, Wyoming, West Virginia, a number of different states now, and in wastewater samples themselves. We've also measured this in zebrafish for specific chemicals and in mice for the lab created mixture. The chemicals promoting these effects and the underlying mechanisms are still largely yet to be determined. These ethoxylated surfactants that I talked about today may be responsible for some of that, but we still have a lot of work to do. And then lastly, there's still a lot we don't know, right? So we still have a poor understanding of human exposures in these areas. We still have a poor understanding of the chemicals used. It's largely self-report, and there's a lot of trade secret exemptions still allowed. There's definitely growing analytical research, but it's still focusing on a small number of chemicals. And then lastly, there's a poor understanding of potential health effects and mechanisms from exposure used in this process. So it's mostly just been screening level toxicology studies and a few really detailed mechanistic studies. And so I'll stop there. I think I went a little bit long. I'll just thank NIEHS for doing all the funding here. My growing little lab, we've got a research assistant and a rotating PhD student, and then a postdoc starting shortly. And then some of the collaborators on some of the work that I shared today. And from there, I will stop and hand it off.
Thanks, Chris. That was fascinating and a little bit scary. You know, I think you mentioned 42% of Americans are obese and another 30% are overweight. And, you know, I think a lot of people think that obesity and overweight is caused by eating too much. You know, clearly there need to be more studies in humans, but I think there's no reason to believe that these studies won't hold up in human studies. And it really is an eye-opener. So thank you very much. I appreciate that. Let's move on to our next speaker, Dr. Laura Vandenberg, PhD, is an associate professor at the University of Massachusetts at Amherst School of Public Health and Health Sciences. Her work addresses how low doses of chemicals during critical windows of development can alter gene expression, cell differentiation, and tissue organization in subtle ways that can lead to adult diseases such as cancer, obesity, and infertility. She is specifically interested in endocrine disrupting chemicals and has worked extensively with chemicals used as plasticizers and flame retardants. Her work also focuses on how traditional toxicology assays have failed to identify a number of ubiquitous endocrine disruptors and how current risk assessment practices can be improved in the study and regulation of this class of chemicals. Tonight, Laura will be presenting A Poisoned Web from unconventional oil and gas production to plastics to endocrine disruptors. Laura, it's all yours.
Thank you. It's so great to be here. I love this metaphor of a web that we've sort of caught ourselves in. And I hope that I can sort of lead you along and show you why, to me, this topic is so important, but also really infuriating. So you've heard already from Chris about this concept of endocrine disrupting chemicals, or EDCs, you'll see on my slides. And also a little bit about fossil fuel extraction, the fracking process, unconventional oil and gas development. But I also want to introduce here why plastics are an important part of this sort of web, or a cog in this story. And so I'm going to use this, this little picture here to sort of orient you to which part of my talk am I talking about. And I'm going to start with, of course, fracking. And again, you know, I think Chris gave a really great overview, but to me, I think the real important take-home message is that a lot of the industries that are involved in hydraulic fracturing, their claim is that chemicals really only make up a really tiny fraction of what is injected into wells, and that's absolutely true. The problem is that when we're talking about tons and tons, it doesn't matter that it's 1%. We're still talking about 1% of hundreds of tons, so or millions of tons. So this is a really important concept to keep in mind, that percentages is a way to sort of hide the depth and breadth of this problem. And I am going to tell you just a little bit about our collaboration with Chris and his PhD advisor, Dr. Susan Nagel. And this work was done by one of my students, Sarah Sapowski, who is now a postdoc at University of Iowa. So these are the same animals that Chris evaluated and found that exposures during early life to this mixture of 23 chemicals increase the likelihood that the females would be obese or overweight. And he saw a frank increase in the weight. That was true also in the males, but we really focus on the females here because we're looking at the mammary gland. We evaluated the mammary glands that were collected from these females prior to puberty and in early adulthood. But today, just for the sake of time, I'm going to focus on the effects that we saw in the mammary glands in early adulthood. So just a brief introduction to the mouse mammary gland, because this is absolutely my favorite organ, and you're going to see why in a second. It's really a beautiful tissue, and it allows us to evaluate the function of a lot of different hormones in the body. The mammary gland is one of those organs that relies on estrogen, progesterone, androgens, growth hormone, some enzymatic activity. And so it really is one of those organs that integrates a lot of biological information. And it's also really important for the health of the species. In humans, we can make choices around lactation, and we also have the option of buying formula, infant formula for babies, either because we choose not to lactate or because we have trouble lactating. But for most of the creatures on the planet, that is not an option for them. So the mammary gland is interesting to us because it's relevant to breast cancer. But it's also really interesting to us because it's important for the survival of the species. The mouse has 5 pairs of mammary glands, you can see here, that lie along the surface of the animal. And unlike a human breast or a cow udder, these tissues are really flat against the skin, which means that we can remove them from the animal and look at them almost in 2 dimensions. So we can look at the whole mammary gland at once. And I'm going to show you what this looks like. This is a picture of the mammary gland that is collected from an animal prior to puberty. The functional part of the mammary gland, the part that will eventually make milk and deliver milk out to the nipple, which is shown here, is the dark purple part. So this is the epithelial cells comprised of ducts. And prior to puberty, it's this cute little tree without very much here to look at. As soon as the animal hits puberty and starts making her own estrogens and progesterones, when her ovaries start pumping out those hormones in really large quantities, the mammary gland and these epithelial ductal structures will undergo very rapid change in development. And that's shown here. So that last mammary gland that I showed you would really fit in just this little part of the entire entire tissue. So in just a few days, a massive amount of growth has occurred in this female. This is a lymph node. Don't be worried. She doesn't have a tumor. So this is perfectly normal tissue. And in the pubertal mammary gland, you can see that at the edges, at the tips of all of these ducts are these bulb-like structures. Those are referred to as terminal end buds. Don't worry about the terms. Really, the important thing is these are sort of like Q-tip shaped structures. And the cells in these structures are dividing rapidly, which allows all of these ducts to sort of squirm their way through the fat pad and fill it in. And the female wants that to happen because if she gets pregnant and produces milk, the more epithelial structure she has to work with, the more milk she's going to produce. And this is just a close-up picture showing you those terminal end buds, those Q-tip shaped structures. And in here, we would see lots of cells that are highly proliferative, and they're also releasing chemicals that allow them to chew apart this fatty part of the mammary gland. This is the adult mammary gland. And I like to use the metaphor of a wintertime tree. So we see ducts, we see these branching points, and they all lead back to Rome. All roads lead to Rome in the mammary gland. In this case, this is the nipple. So eventually, if this mammary gland was promoted to produce milk through hormones, that milk would be pushed down through these ducts, which are acting like pipes, and would be brought to this nipple where the pup would suckle. Again, lymph node, don't worry, not a tumor. This is the pregnant mammary gland. And so you can see the difference between here and here, not a size difference. This is really a difference in the kinds of structures in the mammary gland, and a close up here. This is the differentiation. So these cells are making decisions at this stage to produce the kinds of structures that are needed to make milk, which is what is happening in this mammary gland, which is a lactating female. So all of these structures out here that look like grapes are the cells that are actually producing the milk. And you can see here's a duct, because all roads lead to Rome. All those pipes send the milk back to the nipple where the pup will nurse. I think I've convinced you that this is the most beautiful tissue or organ you could possibly look at least in a mouse, and it is absolutely my favorite because there's so much to see and to explore with our eyes, which makes it very easy to understand whether environmental chemicals are interfering with the development, with the health, with the function in the case of lactation, as well as by promoting disease in this tissue. We can visibly see it. So when we collected those mammary glands from Chris and Dr. Nagel, they sent them to us, we stained them, and we looked at them, and you can see these are adult mammary glands, so they have that wintertime tree looking appearance, just ducts and branches. What we saw is that in the animals that had been exposed to those mixtures, those 23 chemical mixtures, that there was actually more ducts present. And that might not worry you on face value. Maybe more ducts means more milk, but it does worry us because more epithelium, more ducts, means potentially more cells that could also be at risk for other carcinogens or other sites where cancers might arise. When we looked at these cells of these mammary glands, so I just took this tissue and I sliced it, so we're looking at it one cell thick, what we saw was an increase in the number of cells that were dividing, proliferating, and a decrease in the number of cells that are dying. So we've altered this very important health ratio of those epithelial cells, again, potentially a risk factor for mammary cancer. We also saw that there were changes in the expression of the hormone receptors. So if you remember in Chris's talk, he mentioned that many of these chemicals can bind to hormone receptors. And when they do that, they can mimic the natural hormones action, or they can block the natural hormones action. We also think that some of these chemicals, and we don't know which ones because we only looked in the mixture, could also be endocrine disruptors because they're altering the expression of the receptors themselves. So this is potentially making the animal hypersensitive to its own hormones. And then what's really interesting, this is the part of the story that really fascinated me, is that in normal adulthood, we have this wintertime looking tree. And yet in the animals that had been exposed to the fracking mixture, we had the wintertime looking tree, but we had these bulb-like structures at the ends of the ducts. And if you remember, I told you, those are the Q-tip-like structures. Those are structures where there's high proliferation, the cells are dividing. We would not normally see those in adulthood. So the fact that those structures are still present here at this age is concerning. It suggests that these animals are potentially hypersensitive to other compounds, other chemicals, and their own hormones that might promote cancers. We don't know yet the answer to whether or not that's actually true. We would need to do further studies. I'm still trying to convince Chris to do the next series of studies with me, but that's, to me, one of the really interesting questions. But when we did isolate those bulb-like structures out of these animals, it turned out that they were precancerous lesions in the mammary gland. So my lab has looked at a lot of different chemicals. I focus, because my interest in the mammary gland is on its sensitivity to estrogen and the role that estrogens naturally play in the mammary gland. So we've been looking at a wide range of chemicals, not just this 23 chemical mixture, but chemicals that are used in the production of plastics, birth control, chemicals that are used in sunscreens and personal care products, chemicals that are used to make plastics really flexible, and that these chemicals are not actually bound to the plastic, so they're really easily released into food or out of medical equipment, as well as chemicals that are added to personal care products and foods because they act as antimicrobial agents. So each of these chemicals has a real purpose, right? Oxybenzone has a purpose. It is a UV absorbing chemical, so it's used in sunscreens. The problem is that these chemicals also have unintended side effects, and that is that they act like endocrine disruptors. In the case of these chemicals, they all mimic estrogens. And we know that those chemicals are released from those products, even when they're used entirely as intended. So, you know, I often have people tell me, oh, but this says it's microwave safe plastic, so that means it's safe for me to use in the microwave. And I have to tell them, no, actually, microwave safe plastic means that it's safe for the plastic. It doesn't mean that it's safe for you. When we microwave plastics, we encourage the leaching of these chemicals from those plastics into our food. The leaching is generally at low levels. But the problem is that hormones act in the body at exceptionally low levels, which means that disrupting them, even with low levels of endocrine disruptors, could have very serious effects on our health. The big issue also with the reliance on plastics is the fact that they don't go away. They are retained in our environment. I'm going to just show you that the effects that we saw with the chemical exposure, the chemical mixture from unconventional oil and gas, we've seen very similar effects with chemicals that are found in plastics and personal care products. This is an example of mice that were exposed during early life to oxybenzone. And you can see here we have more epithelium. And in fact, we're starting to get those sort of buds on this wintertime tree, which is an abnormality. We've also seen the same thing with animals that have been exposed to bisphenol S, which is a BPA replacement chemical widely used in plastics. And in fact, the only other time that I have seen those bulb-like structures in the mouse mammary gland are in mice that we had exposed to low levels of bisphenol S during early life. So I started by telling you that I see this as sort of a giant web that we're all caught up in. And I want to explain to you what that means and why this fills me with primal rage. And I imagine many of you feel similarly, just fury and anger over this situation that's been created. All of that oil, all of that natural gas, it is going towards a purpose. And that allows some excuses to be made for why we continue to do what we've been doing. Oh, we need this oil and gas. I think one thing that people are not realizing is that that oil and gas is used to heat our homes. It's used as fuel for our cars, but about 10% of it is used in the production of chemicals, including pesticides, and about 5% of it is used in the production of plastics. What this means is that when we drive better cars, When we get better at heating our homes, when we start switching to renewables, the industry is not going to turn off this pipeline until we make them, because there are things that they can make with the oil that is extracted from the ground. And when we think about it, one of the most profound things someone ever said to me was that, we buy these water bottles. We buy them all the time. You should not think about that as I just bought a bottle of water. You should think about I just bought plastic, because the water inside is just a tiny fraction of the entire waste that went into producing even a single water bottle that you really were only using once. Actual forecasts that have been put out by the by oil producers is that over the next 20 years, I'm going to show you the zero line on the graph is right here. So over the next 20 years, they think that we're not going to be using fossil fuels. We're going to reduce our fossil fuels for power and for building. We're going to increase our fossil fuel demand for the production of plastics. So we're doing great things by making our homes more efficient. We're doing great things by putting solar panels and using wind farms and geothermal heating. And we're just going to be diverting those fossil fuels into the production of plastics. And the industry is the one that's telling us that that's what we're going to be doing. So this is a great figure from that report that Ned mentioned at the beginning of our call. And I put a link to the report in the chat box. It's free and it's meant to be easily read. What you can see is we're right here and you're looking at the total global plastics production. This is, again, dire predictions that are being made that over the next 20 and 30 years, there is going to be an exponential production of plastics around the world, which is going to be great for the industries that are going to be marketing this stuff to us. And if we do nothing, if we continue with business as usual, a lot of this plastic is going to end up as mismanaged waste. And very little, I think this is a very generous estimate, very little of the plastic waste that's created will end up being recycled. So I love this quote. On a previous talk that I gave to another group, I misattributed it. So I'm going to properly attribute this quote to our colleague, Dr. Jane Munke, who's the director of the Food Packaging Forum, which is that recycling is the fig leaf of plastic production. And if you don't get the metaphor here, the fig leaf is this tiny little thing that's meant to cover up, something that maybe is interesting. As an endocrinologist, there's something interesting there, but it's this tiny little coverage, and recycling is the fig leaf of plastic production. That's shown here. So this is the production of massive amounts of plastics every year. All of this really derived from fossil fuels. Very, very, very tiny percentages of this are made from biofuels. And here is the amount that's actually being recycled. It's so little as to be horrifying or embarrassing. And we know that as we deal with plastic waste, a lot of it is being incinerated, which means that we're essentially burning fossil fuels in a different way and releasing that waste into the air. And a lot of that plastic waste ultimately comes back and contaminates wildlife and contaminates our own food webs. We know that EDCs pose major risks to everyone, but that there are specific stages of our life where we're at heightened risk for the effects of endocrine disrupting chemicals. So during fetal development is the period where all of our organs are developing and building connections with each other. That inter-organ communication is really essential, and it's dependent on hormones. So even modest changes to the function of hormones in our bodies during those periods of time can have serious impacts on us. I love this metaphor. I live just outside of Boston. If you go get on a boat in Boston, and the boat is pointed towards England, and you say, oh, it's totally fine if the compass is off by 3 degrees, 5 degrees. It seems like a really small difference when you're close to Boston, but by the time you reach Europe, you're going to be speaking French. So small changes can have really big impacts on development. And what we're learning also is that the periods of life when we're sensitive to endocrine disrupting chemicals, it's not just early life. These periods of life continue to sort of impose upon us. And the mammary gland, my favorite organ, develops in early life. It develops again at puberty, develops at pregnancy and lactation. It's also after lactation when you wean your babies. And it also has sensitivity during older life, during aging. What we know, and Chris and I didn't have time to talk about it today, but we know that people who live near unconventional oil and gas extracting facilities are at increased risk for a bunch of different health conditions. If what is being produced from that unconventional oil and gas, which is plastics and other chemicals, is also harming their health, they're getting a double whammy. And that should infuriate all of us, because this ultimately it's not just a scientific question. It's a question of injustice. It's a question of disparities. It's a question of whose health matters. It's a question of whether or not, what's the bar that we set at proving those effects before we're willing to sort of take a stand on them. All of those things are things that personally keep me up at night. And I'm going to guess they keep you up at night too. So thank you for listening. Chris and I would be very happy to answer all of your questions. I think Ned is going to handle the Q&A session. So please, if you haven't already, put your questions in the chat box and we'll stay and answer them.
Thank you, Laura. That was fascinating and infuriating at the same time. Thank you, Chris. We have a few questions coming in, but while they come in, and I encourage everybody to put a question in the chat box and we'll try to get to them. But I did have a question maybe for both of you. And, you know, we often hear the old saying from Paracelsus from the 16th century, the dose makes the poison. What Paracelsus was really saying is that everything can potentially be a poison, even water and oxygen, if the dose is big enough. All things are poison, he said. Nothing is without poison, but the dosage alone is what determines whether something is harmful or safe. Well, we know that things are a lot more complicated than that. Every pediatrician taking care of newborns and small infants and young children understand the fallacy of Paracelsus' statement. Can you explain, can either of you or both of you explain why the dose makes the poison doesn't always apply to the chemicals you're studying in your labs?
Yeah, I'll start. I see Chris laughing a little bit because he knows this is one of my favorite things to talk about. So I think that there's a couple great examples, right? We all know that the more cake that I eat, the more weight that I will gain, right? Like that's Paracelsus. It makes sense, right? Like I eat more cake, I will gain more weight. I drink more formaldehyde, I will feel worse, right? Those totally make sense when we're talking about toxic things. But I like to remind people that example of water is a great one. You can overdose on water. In fact, there's been some really terrible stories of fraternity hazing where young people have died from consuming massive amounts of water in short periods of time. Water can be toxic. We also know you die if you don't have water. So it's a great example where both too much and too little is bad for us, is toxic for us. The same is true for some vitamins and natural elements that both too much and too little will kill us. They'll kill us in different ways, right? A deficiency is going to kill you differently than taking in too much. With hormones, if you can, take in too many hormones. There's a reason why when you're prescribed birth control, they don't tell you, take all of this at the beginning of the month and you'll be totally fine. Like it'll space itself out, right? Like we prescribe hormones at doses that are intended to have certain effects. And we wouldn't give people massive amounts of hormones because they will have uncontrolled bleeding, they will have pain, they'll have signs of toxicity. So hormones are a great example where too little will have a bad effect, and too much will have a bad effect. So this is what Chris was really referring to as if you were looking at a graph, and along the bottom of the graph you were looking at a dose, and along the top of the graph you were looking at an effect, you would see something that looks like a U or an upside down U effect. You really want to be somewhere in the middle when it comes to hormones. With endocrine disruptors, no one gets to choose how much they're exposed to. You can't say like, well, I'll take a little bit of BPA today, because you don't know where you're being exposed to it most of the time. I don't think any of us would say, I'll take a little bit of, a toxic pesticide. That would be silly for us to do. And yet we're doing it all the time in human populations. And those little bits are putting us right in the range of where these chemicals act like hormones. They're not toxic. None of us are dropping dead from drinking out of a plastic water bottle or eating, conventionally farmed foods that have pesticides on them. Unfortunately, for the most part, people are not sick enough in the areas around unconventional oil and gas operations so that we can easily point to it and say, see, those people are being poisoned. It's not that, it's more insidious, and that's why it's scary.
Chris, anything to add?
Really not a whole lot. That was, that pretty much covered it. I would just say I'm taking great joy in the last year or so in getting to give kind of foundations in toxicology lectures to students and telling them that Paracelsus was wrong, and that he also used an awful lot of opium. And so we should not be surprised that maybe all of his ideas were not entirely on point.
That's good to know. You know who likes to quote Paracelsus are the lawyers for the oil and gas industry. They love that.
Tell them about the opium, Ned, maybe.
I have a question here, and I think a lot of us probably wonder about this. I wonder about how all these chemicals tie into the rise of PCOS, polycystic ovary syndrome, that we see in the clinical setting. And we talked about obesity on the rise, and what could be causing that, and thinking, well, maybe it's endocrine disrupting chemicals. And obesity, by the way, isn't just an American epidemic; it's an epidemic everywhere in the world. And what's been happening? Plastics and petrochemicals, oil and gas extraction happens everywhere. But also, we've seen over the past couple of decades, along with obesity, endocrine-mediated pediatric medical conditions. Children under the age of 5 are particularly vulnerable to the effects. I think, Laura, you said that, to even minute doses of these chemicals, of endocrine-disrupting chemicals. We've seen an increase in male reproductive problems, undescended testicles, hypospadias, some testicular cancer, early female puberty, leukemia and brain cancer, which appear to be mediated by some degree by hormones, neurodevelopmental and neurobehavioral disorders, which appear to be on the rise here in this country, at least. Preterm births and secondary consequences from those preterm births are up. Childhood obesity is on the rise. But other than the global surge of chemical production and plastics and endocrine disrupting chemicals, is there another plausible explanation besides endocrine disrupting chemicals that would have such profound effects on reproduction, growth, and development, or on polycystic ovarian syndrome? This all seems to be happening at the same time. And I think the thing that is so fascinating from this discussion here tonight is what else has been going on. And we've seen the rise in plastic and endocrine disrupting chemicals. Shouldn't every parent be freaking out? I guess that's my question.
You just had like 3 questions. Chris, do you want to start with PCOS? Is there any evidence for PCOS? I know that you looked in the rodent at some ovary.
Yeah, honestly, I don't know the literature on this super well. I do know that certainly, BPA has been associated with so many things, but I do know that researchers have found elevated BPA associated with PCOS. And I'm sure a range of other endocrine disruptors. I don't know a lot about the the science of PCOS, so I couldn't warrant a lot of guesses as to like what the mechanisms might be for that. But I mean, I would say generally speaking, if there's a hormonal mechanism in the body that will contribute to some sort of adverse health outcome, there's probably an endocrine disruptor that will work through that mechanism. They are diverse and plentiful.
So I think that Ned's second question was really getting at this issue of like, what signals in the environment could be triggering these kinds of effects? And Chris presented really one of the first seminal arguments that chemicals might be involved in the obesity epidemic, which was really, it was a very large scale look at how many people are getting to be overweight. At the same time, look at the chemical production. But that kind of study is at risk for a bunch of different fallacies, right? So it doesn't mean that the people who are becoming more obese are necessarily in the places where those chemicals are being produced or are more heavily exposed. And that's okay. That was the very first hypothesis. They were asking a question in that study. There are now really well done epidemiology studies that have looked at people before the disease is onset. They've measured how much of these chemicals are in their bodies, and then they sit and they wait. And they look to see, okay, if your mom had more of chemical X in her urine when she was pregnant for you, and now we wait for 30 years, are you at increased risk for different diseases? Now, what I just described for you is a standard epidemiology study, but it takes decades. We're essentially saying, okay, we're gonna let a generation or 3 be exposed to these chemicals while we wait for the data. Most of us aren't so happy with that, which is why we work with animal models as a way to sort of draw tighter lines between what possibly could happen in human populations. But even with all of those sort of caveats of the limitations of human studies, there are now hundreds, at least, I mean, for BPA alone, there's more than 150 epidemiology studies suggesting if you have more heavily exposed to BPA, you're at increased risk for a bunch of different things, a lot of different things, hormone related things. That's just one chemical. Here, we're talking about dozens, hundreds of chemicals. And for the most part, they're poorly studied, but those that are studied, it's just a matter of time before, if it's an endocrine disruptor, before we uncover a health connection. So that leads me to the third point that you made, Ned, and I saw Lois was making this in the chat box, which is, if parents knew about this, wouldn't they be infuriated? And I think in general, I have seen personally 2 different reactions from just general people. Number one is fury, which is how I feel every day. And the other is hopelessness. Right? This problem is so big and it's so out of my control. And you're telling me that it doesn't matter what I do, this stuff is in my water and it's in the air and it's in my neighborhood and it's on the foods that I'm buying without me seeing them. And that can feel very hopeless. And this is where we really need advocacy groups to, number one, show people what they really can do. Number 2, fight for change when it's needed, like at the local level, to say, I don't want this refinery here. Or really, it's not just I don't want it here, I also don't want it in those poor people's neighborhoods either. Like, this is not okay. And number 3, to call for change from our elected officials, because we can do better. We know these chemicals are harmful. So we can demand better from the agencies that are intended to protect us.
Thank you. Thanks. And thank you for getting Lois's comment in there, because I think parents, the more they learn, the more they're concerned about what's going on. Just briefly, there's something in my reading that I think is very important to mention, is that we think that if we're exposed, we individually have bear the risk of health outcomes, bad health outcomes. Can you explain how exposure to endocrine disrupting chemicals during pregnancy can have multi-generational impacts down the road to the mother, to the offspring, to the offspring's offspring, and so forth?
Yeah, so I mean, and Laura can step in here with anything, but I mean, I think that's something that it's still newer science, right? So I think we understand pretty well at this point how those things can directly affect the mother, right? The offspring, the child, they are in early development depending on very precise hormonal cues to develop all sorts of organ systems and bodily maintenance processes and all of those things. And so disruption, even in particularly in early development, can really dysregulate those processes and cause all sorts of adverse health effects that might be lasting. When we start talking about effects even beyond that, right? So we get into this whole question of multi-generational versus transgenerational, right? And so exposure to a mother is often going to expose the mother, to the child who is growing during the pregnancy, and 3, to the germ cells of the child who will eventually become the next generation, the grandchild, right? And so you've got effects at 3 generations just through one chemical exposure. And so that's one of the ways that you can get these multi-generational effects. There's definitely been growing research now showing these transgenerational effects, which are effects even beyond that generation. And a lot of that gets into epigenetics and essentially changes to how DNA is expressed that might be lasting changes inherited by the offspring and the offspring's offspring. And we are starting to see some of that borne out in more highly exposed populations where we're seeing effects now at the great-grandchildren level.
Yeah, I think it just points to the fact that pregnancy is such a vulnerable time because it can affect the pregnant woman. It can affect the fetus as far as the growth and development of their organs and their tissues, but it can affect their germ cells too, that are going to produce, when they mature, their sperm and their ova. And that is, it's like something that keeps on giving, that we don't want to keep on giving. It's really important. I think it's an important point, the multi-generational impact of these chemicals. We need to be aware of that, that it's not just us, that we're just not the only ones taking the risk. It's kids as well. We have a couple of minutes left. I do have a couple of questions that I wanted to ask. Let's see here. I kind of want to get back to fracking because the chemicals used in fracking, the chemicals that end up in the waste are just, there are studies and studies showing that the chemicals are harmful. Not just the heavy metals, not just the radioactivity that comes back in the flow back and has to be disposed of, but the fracking chemicals themselves that end up being shipped in brine trucks or dehydrated to go into landfills or injected in injection wells. And so, there have been a lot of studies looking at health impacts from fracking and from exposure to the fracking pollution. Probably the most relevant studies to talk about here are the birth outcome studies, linking proximity to unconventional oil and gas operations to increased risk of low birth weight, prematurity, small for gestational age, newborns, birth defects, and developmental delays. And can you link those? We won't talk about the respiratory and the heart disease and all the other impacts that are being studied, the cancer. We have a cancer crisis here with pediatric cancer here in southwestern Pennsylvania. And, investigators are looking to see whether fracking has anything to do with that. But when it just comes to the birth outcomes, do you see a parallel between what you're finding and what researchers are finding about endocrine disrupting chemicals and what epidemiologists are finding that's happening to people out in the gas patch?
Yeah, absolutely, I think is the easy answer to that. I mean, we can do a lot of manipulations in rodents as studies to be able to understand what the impact is, and also sort of the exact when question, right? So we can take a mouse and expose them only during this trimester or the equivalent of a trimester, only during this period. And yes, I mean, we can cause preterm birth. In fact, I have a study that's ongoing in my lab right now, and the animals are all mixed up. So I don't know who's exposed to what, but we've seen some of the earliest births that I've ever seen in mice in an ongoing experiment in my lab. So we're able to get preterm birth. We're able to see early death, so neonatal death. We're able to see small for gestational size. So, they're born smaller. And then we also see exactly what Chris talked about, which is the catch-up growth. You're born small and then you overeat. And so you make up for it later on. So the tiny guys end up to be the ones that have the largest body fat later on. So absolutely, we can make those connections in rodents. I think the tricky thing is, and what really makes, the study that Chris and Susan Nagel did, and it was really remarkable for its time, was looking at a mixture, where most of us are looking at a chemical at a time because that's what we're comfortable with, and it's what funding agencies will fund, when in reality, people are exposed to this slew of chemicals. And I saw in the chat, there was a question about, well, what about inhalation exposure? Yeah, absolutely. I mean, these things are out in our environment, and there are plenty of things that we breathe in that also act as endocrine disruptors. So there's no way that I would say, oh, that's the right way to dispose of these nasty chemicals. There's no right way to do that we could say will not pose harm to at least some people, whether it's the workers or the people who live around those disposal sites, or the people who have those trucks sort of pass through their neighborhoods.
Great. You know what? We are running out of time. I just have one more quick question before I turn it over to Ryan. This question is for both of you, if you'd like to take it. Both of you do amazing research. How do you counter arguments from contrarians and from skeptics that you can't prove cause and effect, that you can't prove that this chemical or this combination of chemicals causes such grave health harms? Now, we don't live in a court of law where evidence beyond a reasonable doubt is required in order to act in a precautionary way. But sometimes, isn't it true that sometimes circumstantial evidence is so great, it can be so great and so compelling that should be enough to act? Am I right about that, or am I off base?
I mean, here's what I would say. Those skeptics are right. They're right on a fundamental level. And in fact, we're scientists, so we're supposed to be skeptical, right? We're supposed to constantly be challenging our views. Our brains are full of dogma. We're supposed to constantly be challenging them. Skepticism is not a dangerous thing. But what you just said is something different, which is no level of evidence will ever prove to me that I should be worried about something. You can make the same argument for smoking, right? There's no proof that smoking causes cancer, but we all know that smoking causes cancer because there's an overwhelming amount of evidence linking X to Y. Now, if you get lung cancer, I can't say that that's because of the cigarette that you smoked on October 30th, 1972, right? And that's the level of evidence that industry proponents sometimes call for. They don't just want you to link that it's fracking exposures. They want you to tell them that it's the mercury that you were exposed to on this day at this time from this product. That's a ridiculous burden of proof, and we would not accept that from tobacco manufacturers, and so we should not accept it from other polluting industries. When I talk about this to my students, we talk all the time about correlation is not causation, and that in science we never prove anything, we only disprove. But I also remind them, when it looks like a duck and it quacks like a duck and it waddles like a duck, It could be a goose, but it's probably a duck.
Anything to add, Chris?
Yeah, I mean, not a lot. I would just say that we can very rigorously test these things in the lab, right? So one thing that I don't know that we talked a lot about today is that hormone receptors are highly conserved, right? So the way a hormone receptor works and the chemicals that it binds in a mouse or another lab or a zebrafish are pretty much identical to how they work in a human. And so we can really well model how these chemicals are going to act by studying, controlled exposures, there's Metabolic again, using animal models and controlling for every other factor, right? Controlling for diet and activity and all of those things. And that provides us a much better way to examine that causal effect.
Thank you. I want to thank both of you, Chris and Laura, for joining us tonight. These were great presentations and a really great session afterwards. Thanks to the audience for listening. I also want everyone to know that Environmental Health Project has created a handout on endocrine disrupting chemicals and shale gas development. I've put that in the chat, and you can find it on the website as well. And lastly, I want to mention that Environmental Health Project is hiring a public health manager. To learn more about that role and the application process, please go to our website at environmentalhealthproject.org. So thank you all very much. I'm going to turn this over to Ryan now for some final words. Ryan?